Hello. Hello everyone. Welcome back to my channel. Hope you are all doing good. And here we come with the third part of the splicing mechanism and the last part of splicing mechanism, that is tRNA splicing. So till now what we have learned, that is what we have learned, is a complete different mechanism from what we will be going to read now. The splicing mechanism, that is spliceosomal pathway, and the self splicing pathway was somewhat similar in the mechanism part except to that of the protein requirement. But in this tRNA splicing is completely different process which we will be going to read. So let's start with this process. Before that, if you don't know what is splicing, what are intron, what is exon, and what is the basic purpose of splicing, I recommend you to please do watch the first part of splicing mechanism so that you get concept clear about what is splicing mechanism and why it is performed. So let's begin with the video without any delay. So let's start. Before that, before the splicing of tRNA, processing of tRNA takes place. What do you mean by processing? Or you can say cleavage of tRNA takes place. Okay. You might be wondering, isn't the cleavage or splicing the same thing? I want to tell you, no, it's not the same thing, because cleavage is the removal of the extra part from the structure, whereas splicing is the removal of introns from the structure. Okay, so there is a difference between a cleavage and splicing. So that's why before splicing, the extra 5' end and the extra 3' end need to be cutted out, as a result of which the tRNA is now ready for the second process of modification, that is splicing process. As you can see in this picture, this is the pre-tRNA structure. Okay, want to just highlight it. This is the pre-tRNA structure and this is the 5' end. Okay, this is the extra structure which is found in the 5' end, and this is the extra structure which is found in the 3' end. So first what we will do, we will take two endonuclease enzyme and we will make a cut in this region. As you can see, the RNase P is a protein which makes the 5' cut, which makes the cleavage in the 5' region of the tRNA. Okay, and next another enzyme, that is D and E and F, that is RNase D and E and F, makes a cleavage or cut in the 3' end. As a result of which, you can see this part is completely removed and we get the 5' and the 3' end. Apart from this structure, there is one more extra structure which is present, or a part is present in the tRNA, that is CCA sequence. And you must know that what is CCA sequence. While I was teaching you about the basic structure of tRNA, I already told you that CCA sequence is known as amino acid site attachment sequence. So the amino acid which comes binds to this sequence and then the further processing takes place. Okay, so this CCA sequence is very important for the attachment of amino acid. So this is all about the processing of tRNA. Now moving to the next part is the splicing mechanism. So what happens, this is the pre-tRNA, the extra part is cleaved out, as you can see in the previous diagram. So what happens, one endonuclease enzyme will make a splicing and form two part of the, make the tRNA into two half, that is 5' half and the 3' half. And as a result, you can see this extra portion I am highlighting is, this portion is the intron part. So this portion is cleaved out and is excised intron, that is 5' OH and 3' phosphate end the intron has, and as a result of removal of these intron part, unusual ends will be formed, that is 5' OH will be formed in the 3' half of the tRNA and 2' 3' phosphate will be formed in the 5' half of the tRNA. So as a result, if due to this unusual ends which is formed in the 5' half and the 3' half of the tRNA, we need to correct that half, otherwise how will this part will be joined? We need to join this part because now intron is removed, but before joining we need to correct this part. We need to first correct this ends, then only the joining can take place. So before that, what we need to do, we need to clear, or we need to provide a correct basis. So now what will happen? First is the CPDS. Okay, cyclic phosphatase is an enzyme with the help of ATP, of course. One more thing I forgot to tell you, that in this step no ATP is required. Okay, while the conversion of pre-tRNA into 5' half and 3' half of tRNA, no ATP is used. But here ATP is required to convert this 3' to 2' 3' phosphate into 3' OH and the 2' phosphate end. Okay, so now you can see here clearly that this end, that is the 5' half of tRNA, which was previously 2' 3' phosphate, is now converted into 3' OH end and 2' phosphate end. Till now also the structure is not clear. Okay, till now the structure is also not correct because we need to modify it more. Why? Because this 5' OH is still remaining. We need to convert it into 5' phosphate. Okay, we always know that that is 5' phosphate end and 3' OH end. So 3' OH end is now present, and here only 5' OH is present. So we need to rectify this again. So what will happen? Another enzyme, that is kinase, of course with the help of ATP, will convert this 5' OH into 5' phosphate end. Now we have corrected the unusual bit, but what about this extra 2' phosphate end? We need to correct this also, because we only need 5' phosphate and 3' OH so that the pairing can take place and the joining can take place, but we have extra 2' phosphate here. So for that also another enzyme will be coming to the place, that is 2' phosphatase. Okay, 2' phosphatase, or you can say NAD phosphatase, will come with the help of NAD, which is converted to ADP. It will remove this 2' phosphate, extra 2' phosphate from here, and as a result the proper joining will take place. For the joining purpose of, obviously the ATP or the ASTS is another enzyme is required for the joining of the ligase enzyme is required, basically is for joining of this two end, as a result of which, after first of all the joining takes place, then this extra 2' phosphate end will be removed. Okay, first of all the ATS and the ligase will come and join. Okay, and then after that only the phosphatase will come and remove this extra phosphate, and now this tRNA is ready, proper modified or spliced tRNA is ready for the further use. So by this process you can see, by this multiple steps, first the processing takes place in tRNA, then the 3', 5' half was created, then we need to rectify this modified part, then ligation takes place, that is joining of the two ends will take place, and then the extra 2' phosphate is removed. So you must have seen by this that the entire process is completely different from that we have read about in the splicing mechanism of spliceosomal and the self splicing. So self splicing and spliceosomal is totally completely different from what we have read about now in the tRNA. So this is all about, so basically the main purpose is to remove the intron, extra intron from the part. So by this we come to the end of the splicing mechanism, that is the 3 mechanism which we have completed, and that's all for today. Thank you and have a nice day. If you have any doubt, you comment, like, share among your friends. Thank you and have a nice day.